in allowing us to take all these samples. As most of you know, field work often presents unforeseen challenges. Here we set up a sampler at one of the cleanouts at the residential facility, a skilled nursing facility, and unfortunately there were shuttle vans that were coming out on this driveway, and the bumpers would bottom out and rip out this strip. It was drilled in with 3/8 inch bolts, but that wasn't even enough. So we had to reposition the monitoring setup there. So there've been some issues like that. And also, not every location or service area can be neatly sampled. As you all know, the collection systems really follow topography. And so if we wanted to sample a specific neighborhood or a specific census tract, the way the sewer flows means that we wouldn't be able to necessarily sample that exact census tract. You know, sometimes we'd only be able to sample a very small slice of it, and if we went any larger, we would get an enormous amount of upstream flow from multiple neighborhoods where there wasn't as much interest for sampling. So in logistics, all the samples we're taking are 24-hour composite samples. At the skilled nursing facility, we're setting the ISCO sampler to take a sample every 15 minutes for a 24-hour period. The monitoring requires us to set up an ISCO sampler 1 day prior with ice. And then we get 10 liters worth of sample, which we mix and pour into these sample tubes, which contain a mixture of sodium chloride, tris, and EDTA, which prepare the sample for qPCR analysis. UC Berkeley has spearheaded a regional monitoring group for wastewater epidemiology called COVID Web. They've done a great job in securing funding and increasing their capacity quite rapidly. So they can now process up to 25 samples a week from just our agency alone, and many more samples from the entire Bay Area. If you're interested, you can go to covidweb.org to learn more about this. And COVID Web is taking samples from agencies in San Francisco, Marin, Santa Clara, Contra Costa, and Alameda County, and then coordinating with county health officials and the California Department of Public Health. So again, we're very fortunate that UC Berkeley find funding for this work. These samples can run upwards of $1,200 a sample, which would get very expensive very quickly, but they're able to cover that through some philanthropic funding that they found. Our only costs are the in-kind costs we have for sample collection, although we are looking for funding to add staffing resources, just because it's difficult to set up and collect all these samples. Everyone that's involved in this effort is just carving out time from their existing duties to do this work. So some key takeaways: COVID-19 does not spread through the wastewater. Efforts to culture live infectious SARS-CoV-2 in wastewater have not been successful. It's not to say wastewater isn't dangerous. There are certainly many pathogens you can get from wastewater, but if you're going to get SARS-CoV-2, if you're going to get COVID-19 at the treatment plant, it's going to be from being unsafe around a coworker. That said, fragments of the SARS-CoV-2 gene can be found and measured in wastewater, and wastewater surveillance is just another tool for monitoring the spread of COVID-19. It's certainly not going to replace clinical case data or hospitalization data. Our sole intent is to just have this be another tool in the toolkit that public health agencies can use. And I'll just say also, with regard to the skilled nursing facility, that's really like a pilot that we're running. We're hoping that it provides useful data to the skilled nursing facility. There have been some kinks in getting that to work out, but we're hoping that it can be learned from and potentially deployed in other places. So that was my presentation. I ran through it a little bit quicker than I was doing in my practice rounds. So I guess that gives us a little bit more time for discussion and possibly a little bit earlier break for lunch. So I see there's some questions in the chat. Brenda Donald says, is this run primarily by academics, or are the universities contracted with the state? So certainly university researchers are the bulwark of the testing going on. There are private labs that are also offering the tests at a cost, of course, and it really varies from location to location. The CDC has recognized the value of this, and they have initiated what they're calling the National Wastewater Surveillance System, and they've actually contacted the State Water Board in California, as well as regulatory agencies in other states, to start collecting this information. I would say, though, that primarily this is being driven by local university researchers and local utilities who are taking the initiative to provide this information to public health agencies. But certainly there is interest from the states, particularly state public health organizations and federal public health organizations such as the CDC. Yeah, so if you guys have any other questions, feel free to drop them into the chat or unmute yourself if that's available to you.
Well,
yeah,
Ben Lavender has a question here. Ben, did you want to ask a question about pepper virus?
Sure. I wanted to clarify what I thought I heard you say. It sounds like the pepper mild model virus is being used as a surrogate for the concentration of human waste in wastewater. Did I hear that correctly?
Yeah, exactly. And so what they'll do is, they're basically dividing the concentration of SARS-CoV-2 genes by the concentration of pepper mild model virus. Just to make sure that, say you had a service area where it was mostly industrial and you just had like 10 people, but you had an enormous amount of flow versus a residential area with many people. That's what's allowing them to determine the strength of the human waste. Yeah.
Got it. Okay, that makes sense.
We have another question here from Catherine Curtis. What has surprised you the most during doing this work? And did you find things that were unexpected?
What has surprised me the most doing this work? I guess, I don't know, it's that surprising, but it just really is, it really drives home just how comprehensive and impressive the collection system is, in that it goes literally everywhere in the city and collects waste from literally every individual in the city. I think just going out to all these different locations and then looking at the GIS folks, working with flows to try and identify sampling areas, that was probably the most impressive thing to me.
Ryan, does this dealing with this help you feel, what's the word I want to use, more significant in the work you do?
Oh yeah, certainly. It definitely makes, I think I would be a little bit bored. Work this allows me to get out and take these samples, and it feels like I'm actually doing something useful, which, yeah, is a benefit to the community. So certainly it's nice to do this kind of work.
Wonderful.
And then I see in the chat, has PUC previously done this type of public health surveillance? Certainly, we test the wastewater for fecal coliform, but never anything where we're looking at new and emerging pathogens. Which is not to say it's other agencies haven't done that. It's been done in other places. Israel has done it to track cases of polio. There have been other agencies that have done it to track cases, opioid problems in their communities. It's also the RNA sequencing has been used to track where there have been releases of untreated sewage into water bodies in places like Chesapeake Bay. So there's a lot of potential with this tool, not just for COVID-19.
Can I ask another question?
Yeah.
Go ahead, Ben.
Is anyone yet sort of thinking forward to using wastewater-based epidemiology as a tool to monitor the success of a vaccination campaign in given communities?
That's certainly one of the things that the researchers have said is going to be a use for this tool is when we're eventually getting to the point where, we want to give the all clear. This will allow us to determine when it's not showing up anymore in our wastewater systems. So it will allow us to determine, there will be some corroborating evidence that indeed COVID-19 spread has ceased. So certainly there's a role there.
Wonderful. If you have any other questions, please put them in the chat, or you can just talk up and speak them. Some time remaining. All right. Ryan, has there been any other big projects you've worked on relative to research that has had something that you can think is memorable?
Sorry, just research in general? Or this
Well,
research?
just something memorable about your sampling that you'd like to share with us.
Yeah, I think the University of Arizona case it's just really nice to be able to see the theories about this type of testing play out with actual cases. So the wastewater surveillance catching that infections in Tucson, Arizona before it actually occurred. That was really interesting to see. Also at University of Arizona, when they were able to determine that there were infected individuals in the dormitories from wastewater surveillance, that was really great to see. I think at the end of the day, we're just hoping that this is just another piece of data that the officials can look at, and hoping that there aren't too many surprises. It just stays nice and boring for public health officials in the Bay Area when they look at this data. So.
Wonderful.
Yeah, thanks so much everyone for listening, and I hope everyone stays safe.
Wonderful. Yes, thank you, Ryan, for providing this great information and sharing your experience and everything. And we want to thank everyone for joining us today. We want to ask again that you fill out the survey that's in the chat, and also you will be receiving an email for the survey as well. It's a short survey, shouldn't take you too long to complete. And also we want to invite you to attend more of our events for the training buffet and even in the future. And just thank you once again for being with us today. And we hope that you have a great rest of your day.